Hi, Alberto, you're back.
Yes, yeah, I had a problem, so I don't know what happened.
Do you want to give it a try again?
Yeah, please. Can
I
you
see your screen. Yeah.
Okay, let's go with that.
Do you want to screen share and quickly check? Yeah, we see it. Perfect.
Okay, well.
Thank you so much.
Oh, thank you. Yeah, I don't know what happened.
Hi, okay. I just lost you guys for a minute there, but can we all hear everybody?
Yeah, and Alberto is able to screen share, so that's good.
Okay, great. So we've just got one more minute until the official start, and I think the live stream on YouTube is working. So we'll have Adam go first because he is less senior, and then Alberto. Really excited to be doing this again. It was nice to have a break, but it's good to see everyone.
You're an addict now.
What's that? Oh, I'm an addict. Yes, addict to sharing wonderful science. All right, just got confirmation live stream's working. Thanks. That's actually my dad, so good to have family support.
That is just amazing.
Yes, no one can say I don't have supportive parents. All right, cool. So in the interest of time, for once in my life, I think I'm actually going to get started on time. I'm sure more people will continue to trickle in. So hello, everybody. Hi, and welcome back to Cell Migration Seminars. I'm really happy to see so many familiar faces and familiar names, but also lots and lots of new people. We've had lots of new people signing up, so it's really awesome that there's so much continued interest. So I think we have a really amazing lineup this fall. I just want to quickly say that we're going to keep this going as long as there's interest. So keep showing up, keep being supportive, keep asking questions, and we'll keep doing this. And we've already got some really fantastic folks lined up for the spring. All right, so with that, we'll get started. For anyone who's new, the way that we do questions here is that you can just leave your question in the chat and we'll ask it on your behalf, or you can use the raise hand function, or you can type your question into just type the word question into the chat and we'll just call on you. And so we'll roughly try to sort of go in order and try not to miss anyone. So before introducing our first speaker, I'd like to formally welcome Ankita Jha. She's our new co-host and co-organizer. So Ankita is a postdoc in Claire Waterman's lab. She also does really fantastic science outreach, and it's really, really great to have her on the team.
Thank you, Jen.
And then thanks, thanks for being here. Awesome. And so that actually brings us to our first speaker, Dr. Adam Shellard, I'll have to say. I actually don't know after a year and a half, which is the right emphasis. Anyway, so many of you recognize Adam because of course he's the co-founder and the co-host of the series, but he's actually stepped back from organizing and hosting. But I do want to say that it was his tweet 18 months ago where he said, "Cell migration seminars, anyone?" That really started all of this. And so I want to take a second and thank him for all of the time that he put in. It's been, a lot of work. And so Adam's been really fantastic. And now I have a new appreciation of a lot of the admin stuff he was doing behind the scenes. So thank you, Adam. Okay, and so now he's our first speaker. Adam did his PhD and he's currently doing his postdoc with Professor Roberto Mayor at University College London. Today, he's going to be talking to us about collective Durotaxis in vivo. And take it away, Adam. Thanks.
Thanks. Can you see my screen and my pointer?
Yeah.
Thanks a lot.
I don't actually see your pointer. Can you wiggle it around or maybe? Oh yeah, I see it.
Wiggle.
Thought it was mine.
That's
all
right. Cool. And yeah, thanks a lot. And thank you for taking over and good luck with all the admin. Yeah, so and please just give me a shout if I'm running too late. And sorry if I sound out of breath because I've just run up some stairs because I realized I was late for this. So I want to talk about our work on Durotaxis. Let's see if this is working. So I'll start with probably the most famous cell migration movie, I guess, from David Rogers in the 50s, of a neutrophil running around chasing a bacteria. I'm sure I don't need to tell you that you can appreciate that this cell is moving in a highly directed way. It's not moving around randomly. And that's because we know that many cells, including neutrophils, undergo a process called chemotaxis, where the cell is being guided by a soluble gradient of chemical Q. Chemotaxis is now really well understood, or quite well understood, I should say, and has been shown for many, many different cell types in vitro, and has been proposed to underlie a lot of directed migration in vivo. But at the same time, we can appreciate that in vivo, there's lots of stuff going on around a migrating cell. So this is just a figure from a review showing some migration of cancer cells. And you can appreciate that if a cancer cell wants to start moving around in an organism, there's going to be stuff in its way. There's going to be ECM fibers, aligned or non-aligned, and different types of ECM. There's going to be other tissues it's going to interact with. There's going to be blood vessels. There's going to be muscle fibers. There's all sorts of stuff in its way. And all that stuff means that the cell is going to be interacting with that stuff. And one of the ways it interacts with it is in a mechanical way. This graph shows the wide range of stiffness, which is one mechanical property. It's the one I'm going to be talking about today, which is exhibited by an enormous range of tissues with orders, many, many orders of magnitude different. So just really obviously, bone is really stiff and brain is really soft. And of course, the heterogeneities in no stiffness is not just between organs and between tissues, but also within tissues and along a fiber and at all different sorts of scales. And cells can respond to the various mechanical cues of their environment in lots of ways, which is illustrated by this figure. And the way I want to talk about today, which is with regard to stiffness, and the way that's generally thought how it works is that if you have this cell, it can contact the extracellular matrix around it via integrins, which are going to bind to the ECM, and that's connected indirectly to the intracellular cytoskeleton, which means that if you produce a force, so if you contract your cytoskeleton inside, you produce a force in the environment, but also that is fed back internally so that if the environment is stiff or whether it's soft, you can actually sense that by the deformation of the substrate. So this idea that there's external mechanics that could have a role in cell migration is not a new one. And one of the ideas from the 90s was that that maybe mechanics could actually act as a guidance mechanism in the same way that chemicals can in chemotaxis. And really the first seminal paper that came out, which was in 2000, was able to achieve that thanks to hydrogels. So in this case, what you can do is make put two solutions down of acrylamide with a bisacrylamide, which is the crosslinker, and dependent on the ratio of those two things and the quantity, you can generate gels of different stiffnesses. And if you put one next to another, you can make a gel which has a gradient of that stiffness. And then this paper came out in 2000 where they put fibroblasts on these gels. And you can see this cell here. This is a fibroblast cell that's on the soft side of the gel, and it's approaching the interface with the stiff side. And it approaches, it makes a protrusion over the interface, and then it walks across. So it likes to go from soft to stiff, but if you take this, a fibroblast that's coming at it from the other side, from the stiff side approaching the interface, it approaches, it sees the soft side, and it doesn't want to go across, so it stays on the stiff side. So this movement going from soft, in general, moving from soft to stiff is what's called durotaxis, which is migration along a stiffness gradient. And then fast forward from this to 2016, the lab with Xavi Trapat took this and then looked at it in collectives. So this is a movie of MDCK cells. The cells are moving from the middle region out into the free edges on either side. And in the top panel, that collective is on a gel of uniform stiffness. And you can see the symmetrical expansion of those two sides. Whereas on the bottom, the the cluster, the sheet has been placed on a gradient gel going from 6 kilopascals to 70. And you can see that this is now asymmetrical with the expansion preferentially going in the direction of stiff. So we're at a point where many of different cell types has now been shown to undergo Durotaxis in vitro, especially single cells and to a lesser extent collectives. But there's still really scarce evidence for whether it can actually happen in vivo. And some of the reasons behind this, I think, are due to the difficulties of doing mechanical stuff in vivo. So first of all, how do you access the tissue that you're interested in a way that doesn't affect the properties of whatever you want to see? How do you measure your mechanical thing in a way that is that you can generate actually a large amount of data, but also is precise? And often those two things are conflicting. And how do you manipulate whatever it is mechanical thing you want to do without affecting many other things and also affecting, for example, a gradient in the way that you want to? So those things are challenges. But if you can do those things, I think there's a lot of interesting stuff to be learned. And so the question I want to address here is whether cells can be guided by mechanical gradients in vivo. So to address this, we've used Xenopus as a model system. Xenopus is a really good model system for mechanics because you can do all sorts of treatments. You can push it, you can squeeze it, you can compress it, you can cut it, you can do all sorts of things. And this is the migration of the cranial neural crest. So these are neural crest labeled cells. And you can see that they're migrating in a collective manner and they're migrating in a highly directional manner from the dorsal side, which is up here, down to the ventral side down here. This circle here is the perspective eye. So one way you can think about why the cells move in this direction rather than any other direction is because of a chemical map. So if you imagine you put all your chemorepellents in the regions where you don't want the neural crest to go, you put your chemoattractants in where you do want them to go, and that's how they can be guided. And that's exactly the case for the neural crest and also for other systems. So in this case, I'm not going to talk about so much about the repellents, but I do want to mention the attractant. So in this system, the attractant is STF1, and it's produced by a tissue that is next to the neural crest called the placode. So the placode is here, the neural crest is here, and this you can see is a double in situ against a neural crest marker and a placodal marker just next to it. And this is just before the start of migration. And this is how, this is kind of an animation that summarizes a paper previously from the lab about how this works. So the neural crest here in green are chemotaxin towards the placodes in pink, thanks to this chemoattractant of STF1. But when the neural crest contact the placode, they actually engage in an end-caterian adhesion between these two cell types, and that causes repulsion of the placodes away from the neural crest. So what that means is that the neural crests are constantly chasing the placodes by chemotaxis, and the placodes are constantly running away from the neural crest because of this repulsion, which is contact inhibition of locomotion, but I won't go so much into that. So if we take that embryo now from a side view, but I'm just going to put a cross section through it so you can see how it looks from the cross section. So this is what's happening. So you have the neural crest, which is attracted to the placodes by this chemoattractant gradient of STF1. And it moves towards the placodes, and the placodes are moving away rightwards on this diagram. And one of the observations we've always known about the neural crest is that they require fibronectin as a substrate in vivo and in vitro. But we haven't actually known what the location of that fibronectin is with respect to these two cell populations before, because we've never managed to get some staining that can label all three. But you can see here that quite clearly the fibronectin is exhibited right in between these two cell populations. And it's a very, very thin layer. So this made us think that maybe the neural crests, are using the ECM, they're connecting to it via integrins, but because there's this layer of placodal tissue, which they're kind of walking on a little bit as the placode's running away, maybe they're using this tissue as a substrate for the neural crest, for the migration. So to have a look at that from a mechanical perspective, we have to do some measurements. And to do that, we've done nano indentation. It's very similar to AFM, but it uses an optical system. So basically, we've got a cantilever with a spherical glass bead on the end, and we lower that, we indent our sample, whatever that is, in this case, it's the placode. And then dependent on the amount of deflection you get from your sample, you can infer the mechanical properties because if the sample is very stiff, you get a large deflection, whereas if it's soft, you get a small deflection. And in our setup, which is an optical setup, there's basically a wavelength of light traveling down here, and the change of that wavelength because of the deflection is how you can infer the mechanical properties. Okay, so now we're looking at the stiffness of the placodes as a potential substrate for the neural crest, and we're measuring the stiffness, and this is now a 2D heat map of the stiffness across the region of a representative embryo across the placode, and you can see that it's relatively uniform in stiffness, and this is at a stage just before the neural crest starts to migrate. But if you look at it an hour later or so, what you find is the emergence of a stiffness gradient. The gradient is oriented in the axis of the neural crest migrating, so from dorsal to ventral, where the regions that are very close to the neural crest, on top of the neural crest, are very soft, and further ahead are stiff. And this happens at the stage when the neural crests begin to migrate. And we can check that this is definitely for the placodes that we're measuring, because if we remove the placodes either by ablation or by injection of a morpholino against ER1, which is a factor that's required for the development of the placodes, you lose the gradient. So here you can see a heat map of, this is now an average of many embryos, and you can see the gradient in control, but lack of gradient when you lose the placodes. Okay, so we've got this emergence of the gradient, but we know that over time, the neural crests are continuing to move forward, and the placodes are also continuing to run away from the neural crests. So we wondered what's happening at later stages. So this is now an average heat map of many embryos at each of these different stages. So these two are what you've seen already, where you don't have a gradient and then you have the gradient emerge. But then what happens at later stages is that gradient persists, but it spatially shifts in the embryo. So essentially on this diagram, the placodes are moving to the right, and so here the gradient is shifting spatially in the embryo. So what it means is that the neural crests are continually pursuing this gradient, which is moving, and the gradient is moving because it exists over the placodal tissue, and the placode itself as a whole tissue is running away from the neural crest. Okay, so just to summarize that, we've got here the neural crest, which we know undergoes chemotaxis towards the placodes, and now we find that there's this gradient of placodal tissue stiffness. So how is this gradient formed? Because the gradient was being found at the exact stage when the neural crests begin migrating, we thought that maybe the neural crest is generating the gradient. So to have a look at whether you need the neural crest, we got rid of it. So in these cases, we've either ablated the neural crest or we injected a morpholino against twist, which is required for neural crest development. And in both cases, loss of the neural crest results in loss of the stiffness gradient. And the second experiment we did is because if the neural crest was really generating the gradient, what we should be able to do is move it into almost the opposite side of the embryo where it would normally end up, and see whether it can produce an ectopic stiffness gradient in a place where there shouldn't be a gradient normally. And that's exactly what we find. So if we measure the ventral region of the placode where there's normally no gradient, and we move the placode down to that underside, we actually get the formation of a stiffness gradient, and that gradient is formed in the opposite direction to normal. So now the soft is ventral and the stiff is more dorsal. So it suggests that the neural crest is generating the gradient. And also, what about the molecular mechanism? I mentioned at the start that in this chase and run model, which we've previously published, when the neural crest contacts the placodes here, they form an end-caterian adhesion that causes the placodes to run away. So we wondered whether that could be the interaction. So we took out placodes, we dissected them out of the embryo and then cultured them on either fibronectin alone or fibronectin with recombinant NCAD on the plate, and then measured the stiffness. And what we saw was a reduction in the stiffness of those placodes when you have end-caterian, which suggests that end-caterian is sufficient to reduce the stiffness of the placodes in the presence of fibronectin. And correlated with that, what we found was through immunostaining of phalloidin, we found a reduction in the total levels of actin. So this is kind of correlated with the idea that the organization and amount of cytoskeleton you have in the tissue and the cells might be a decent readout of the stiffness. And now finally, we tried a rescue experiment to see whether this was really true. So here we have a control embryo, and now we've knocked down that end-cadherin interaction between the neural crest and placodes. So we lose the migration of the neural crest in vivo. And this is the stiffness gradient we would normally have in the control, and the loss of the gradient we have in the NCAD case. And that's a quantification of the migration. And then what we did was push down on the embryo locally in a region ahead of where the path should normally go in these NCAD knockdown embryos. And what we saw was a rescue in this migration. And if we look at the stiffness in these rescued ones, what we find is that the pushing is actually generating an exogenous stiffness gradient. So we're able to rescue the migration of the neural crest in vivo in NCAD knockdown embryos by applying our own exogenous stiffness gradient. Okay, so just to sum it up, we've got this stiffness gradient, and we think that the stiffness gradient is through the interaction of NCAD between the neural crest and the placode, which generates a local softening at the rear of the placode, and therefore the front of the placode, which is untouched, remains stiff. Okay, so we've got this gradient. The next question to ask was whether the gradient was actually doing something or not, or whether it was just a nice thing to look at. So to do this, we needed to get rid of the gradient. And to do that, we used a similar technique to that which our lab has previously described. So here is a top-down view of an embryo with the right-hand side and left-hand side, with the neural crest here and the placodes just ahead. And what we did was an ablation of the ectodermal tissue away from that region. So the way we're thinking about it is that if the ectoderm is this continuous sheet across the embryo, if you make a cut, then you're just kind of relieving the tension across that tissue. And when you do this technique, you lose the stiffness gradient, which is shown by the pink values. And the result of that is a loss of neural crest migration in vivo, which you can see from the in situ and a quantification on the right. And we can also do some live imaging in these cases. So the left is a kind of a low magnification image showing normal a graft of the neural crest in normal migration, and in the case where the stiffness gradient is ablated. And the right just shows some individual cells moving about in these two conditions. And some quantification of that, obviously the directionality is clearly lost in these cases. But interestingly, we don't find that the speed of the cells is changed, which indicates that the gradient is for the directionality rather than general motility. Okay, so the gradient is used for neural crest durotaxis. And just one thing I haven't had time to tell you about, but we know that this mechanism of durotaxis absolutely requires integrin. So if you knock out integrin, you don't get durotaxis. Okay, so the final thing I want to talk about is whether there's interplay between these two mechanisms. So we've shown previously there's chemotaxis, and now there's hints that there's durotaxis. So is there interplay between these two gradients in the embryo? So to test this, we manipulated these two gradients. So here you see a control embryo with the normal neural crest streams. And if we get rid of SDF1 using morphlino, we obviously lose that, which can be rescued by implanting an SDF1 coated bead into the embryo. So here we're just rescuing chemotaxis. But now in this condition, if we do the stiffness gradient ablation, we then lose the neural crest migration, indicating that the chemotaxis in vivo is dependent on durotaxis or on the stiffness gradient. And then in a second set of experiments, we decided not to manipulate with the endogenous gradients, but actually just to put our own ones on there to compete. So here you can see an embryo with the neural crest streams. And in this case, what we've done is place an SDF1 coated bead into the opposite direction to that which it would normally migrate. So this would be an exogenous chemotactic gradient, and additionally push on that bead to generate an exogenous durotactic gradient. And hopefully you can see that the stream is now moving in the opposite direction. And so this image shows the overlay of these two sides of the embryo, the cyan and the purple, and this is just a diagram of that. And this graph shows the quantification of that. One thing I haven't had time to show you is that if you do either of these two techniques alone, so either pushing or the chemical gradient alone in the opposite direction, we seem to be unable to get that ectopic migration, and you can only get it when you have both gradients. So just to sum up, this is an idea where you have this kind of interaction between these two tissues in both a chemical and a mechanical perspective. So the neurocrests are not only chasing the placodes by chemotaxis, but also are engaging the neurocrests, which engaging the placodes, which cause the placodes to be repelled away, as well as generating a stiffness gradient on which they could follow thanks to this fibronectin that exists in between the two tissues. Okay, and just to finish up, that's all my results. So just to say that I think that durotaxis may be more widespread, and mechanical things could be a generally guided mechanism in vivo. I just want to mention two other papers. So the first is from Christian Francis in Cambridge, and what they showed is that previously, much like our system, it was believed that there's plenty of evidence that axonal turning and guidance, in this case the Xenopus eye, is based on a chemical map. But what they found in 2016 was that there's also a stiffness map. So this is a AFM map of the stiffness, and the blue indicates the axon being guided to the right here. And what they found is that it's actually guided towards softer regions and being turned away from stiffer regions. And just a second example to mention is that a paper last year came out showing, describing the morphogenesis of the mouse limb bud, which involves a movement of cells along this kind of arrow. And what they found was that the movement of that cells correlates with a gradient of fibronectin, and that also is mapped onto a stiffness gradient. So there's a stiffness gradient being exerted here. Whether that's durotaxis, haptotaxis, all these things remains to be seen, but I think there's decent evidence here that mechanics could be involved. So thanks to my lab, especially to Roberto, who supervised me, and yeah, I'm happy to take any questions. Thanks for listening.
Awesome, fantastic, Adam. I'm so happy that we got to see your story. So we have a couple questions in the chat, and people should keep dropping those off. So the first one from Radhika Atit asks, well, says, thank you for the series and all your efforts, agreed, and asks, if integrins are absolutely required for durotaxis, how do you separate this from haptotaxis, gradient of adhesion? Do you have any assays to really tease apart durotaxis versus haptotaxis?
So if we look in vivo, we've quantified the fibronectin along the axis of migration, and we don't see anything that suggests that there's a fibronectin gradient, which is what you would normally think of for haptotaxis. But yes, integrins are involved in, integrins are required for mesenchymal migration. So it's not just like integrins, the only thing they're doing is for durotaxis, they're required just to adhere, and therefore for everything else. So no, I'm certainly not saying that integrins are only used for durotaxis. They're used there, but they are required for durotaxis. There may be haptotaxis additionally going on in the system, but we see no evidence that fibronectin is existing in a gradient. And then in terms of the chemical, we don't know whether the STF1 is being received as a soluble signal or whether it's binding to the fibronectin as a gradient. So that also could be a haptotactic rather than a chemotactic mechanism.
Thank you, Adam. There's a question in YouTube from Abhishek Das. Is the placode acting like a point source of signaling here, whose gradient is diffusing in all the direction, or is it linear gradient in space? And does that matter in tissue scale?
Yeah, so we see it as a linear gradient, so only along that axis. I'm sorry, what was the second part?
Sorry, let me go to that. So second part is, how does that matter in the tissue scale?
How does that matter? I mean, it matters for the neural crest to have a gradient along its axis. So if there was a gradient in the other direction, it may not care, but it wouldn't be received. So at least the current understanding of what durotaxis is you have a cell or a cluster, and then because you've got integrins across that thing, you can pull on one side, pull on the other side, and then you get different mechanical reception from those two things. So as far as I can understand, as long as there's a difference along the axis in which the thing is moving, then that's what matters.
Right, great. We have a question from Ava Pillai, if you'd like to unmute and ask your question.
Hi, Adam. Great talk.
Hi, thanks.
Super cool. But I was wondering, you see an increase in end coherence. So I'd expect then that you've increased adhesion or high cell binding, and therefore I'd expect higher stiffness. So, but you actually find that there's a lower stiffness, right? So could you speculate why that's happening?
Why the stiffness is low when you have NCAD?
When there's more coherence, yeah. I thought that was very interesting.
Yeah, so don't know is the short answer. We know that there's a lot going on, because when the neural crests contact the placodes, there's contact inhibition of locomotion. And so we know that there's a repolarization, that there's loss of focal adhesions, first of all, almost like immediately, that there's a massive change in the actin cytoskeleton, because all of the protrusions that are directed towards each other need to disassemble themselves and then polarize in the opposite direction. So there's a lot of cytoskeletal changes, and there's a lot of loss of focal adhesions. So we think those are reasons to think that stiffness will change. Why it's softening rather than stiffening, I have no idea.
Cool, thank you.
Yeah, thanks.
That sort of answered Tony's question, which was next one. I'll take Yutaka's question. Does each single cell detect the stiffness gradient, or a cohort of cell needs to work together to detect a gradient, which is too small to make sense in short distances for a single cell diameter?
Yeah, we've done that in vitro, and in vitro, the single cells don't respond on a gradient, but collectives do. But I haven't done the experiment, which I think would work, which is if you put a really steep gradient, the single cells might be able to respond. So, I mean, I know it's been shown for chemotaxis and also for galvanotaxis that in both cases, collectives are better than migrating along the gradients than single cells. And actually in the Tropat paper I mentioned earlier, they did that experiment, and they also saw that collective cell Durotaxis is more efficient than single cell Durotaxis.
Thank you very much. Great talk.
Thanks, Yutaka.
Great. Okay, so we're not going to be able to get through all of the questions that are in the chat, but we'll do a couple more. So from Sergey asking, in the opposite direction, does the softening of the placode by the neural crest affect the placode migration?
In the opposite direction, does the softening of the placode affect what, sorry?
Is the placode migration affected by the neural crest?
Don't know. We know that the neural crests migrate. I didn't show that data, but on the embryos where you don't interfere and start measuring the stiffness, the neural crests migrate opposite direction. So I suspect that the placodes should also move the opposite direction, but I don't know.
Okay, so one more from Shrija. She's curious if the fibronectin gradients were images before and after crest migration.
Fibronectin images before and after neural crest migration.
I think the image you showed, yeah.
Oh, what was the image? Oh, the image was near the start of neural crest migration. So normally you have the interface is like this, like neural crests are sat next to placode, and then once they start migrating, they'll start like kind of crawling out. So you get that interface of one almost looking like it's on top of the other, but they're really both moving. Yeah.
All right, so I think this next one will be the last question from Eileen Gentleman. Asks, what are your thoughts on why the in vivo data seem to suggest cells migrate towards soft, but in vitro experiments show that cells migrate towards stiff?
So maybe I didn't communicate properly. So our data suggests that they move towards stiff in vivo. So the gradient
so you're
is,
in agreement.
Is that, yeah. So maybe the difference was that in a paper I mentioned from Christian Francis' lab with the axons, they turn towards soft. So that's a difference. So in our system, it moves towards stiff. For the axons, it moves towards soft. Don't know. I know part of the axonal turning is to do with piezo in their system. I have no idea if it's involved in ours, but in general for Durotaxis, cells move from soft to stiff. In almost every paper, I think in almost every paper, apart from one preprint that's been studied for cells migrating in vitro, they've always gone from soft to stiff.
All right, fantastic. So maybe we'll stay afterwards and answer some more of these questions, or maybe folks can email you. But thank you, Adam, so much for that fantastic talk. And we're all really looking forward to your paper coming out soon.
Okay, so our next speaker is going to be Alberto Elosegui Artola. He is currently a group leader at Francis Crick Institute, and he's also a lecturer at King's College London in UK. His lab seeks to understand how physical properties and biochemical cues work together to regulate biological functions. And today he is going to talk about the role of extracellular matrix viscoelasticity in tissue and the spatiotemporal organization. So over to you, Alberto, and thank you for joining us today.
Well, thank you. Can you see my slides and the
Yeah, we
pointer
can.
tool? Okay. So yes, so hello everybody. First, I want to thank you for giving me the chance to present this project. I will switch gears a little bit from what Adam has presented. Instead of looking at the stiffness, I will talk about another mechanical property, the viscoelasticity. And I will explain the project that we have been working on for the last years, which is trying to understand the role of the matrix viscoelasticity in tissue organization. So most of our research is trying to understand how the mechanical properties of the extracellular matrix regulate cell and tissue response. As you all know in this audience, like tissue rigidity, the stiffness of the matrix regulates many biological processes in health and disease, from morphogenesis to cancer. And I think that a good example of the importance of the mechanical properties nowadays can be appreciated in this mammogram, as tumors are diagnosed by detecting changes in matrix density or also by tissue palpation. And among the different mechanical properties of the matrix, research has overwhelmingly focused on the influence of the stiffness, the elasticity of the matrix as the key determinant of tissue response. Here you can see in this seminal work from Valerie Weaver's lab how they seed breast cells in a soft substrate that they form what resembles a healthy mammary duct. And only by increasing the stiffness of the substrate, these cells acquire a malignant phenotype. However, extracellular matrix and tissues are not purely elastic materials. They don't behave as springs. Extracellular matrix and tissues are viscoelastic materials. So we have developed these videos to show you the differences between elasticity and viscoelasticity. So on the left side, you have a polyacrylamide gel. Polyacrylamide gels behave as purely elastic materials and are widely used in research to mimic the extracellular matrix properties. So when you remove the load from a purely elastic material, it immediately returns to its original position. And in these elastic materials, the initial and the final positions are the same. There are no permanent deformations. And on the right side, you have a brain. A brain is a viscoelastic material. And when you remove the load from a viscoelastic material, there is not an immediate response. There is a time-dependent progressive recovery. And also you can observe that there can also be permanent deformations. So the initial and the final positions can change. So there have been several recent papers that they have shown how the matrix viscoelasticity regulates single cell behavior like cell spreading, migration, or differentiation. But this project was more focused on trying to understand the role of viscoelasticity in a collective response. So how it affects like tissue response. So to develop the project, we wanted to develop a material or use a material where we could control the elasticity and the viscoelasticity independently and control them independently without changing the pore size or the ligand density. So if you think about most research developed in 3D, we use matrix gel, collagen, or fibrin, these natural matrices, and all these mechanical properties all these parameters are dependent. So for example, if we want to increase the stiffness in these matrices, what we usually do or commonly use is we increase the concentration. And when we increase the concentration, we are also decreasing the pore size and we are increasing the ligand density. So it is impossible to decipher what is the parameter that is driving this response. So we have been working with alginate, that with alginate we can control the stiffness by changing the amount of the ionic crosslinker. And we can also change the viscoelasticity by playing with the molecular weight of alginate. And all this without changing the pore size or the ligand density. So we started the project with two matrices that had the same stiffness, but they have very different viscoelasticities. So you can see on the graph on the right side that the stress relaxation time is very different. The stresses relaxes much faster in the viscoelastic matrix. All the work that I will explain is related to a work that it has been submitted. And if we look at this is a common experiment that we have performed during this project. So these are MCF10A cells. These are non-malignant breast cell line. And these are spheroids of about 2,000 cells that are encapsulated in 3D in matrices of the same stiffness. So if you look at these videos, the only difference between these two matrices is their viscoelastic properties. So on the left side, you have a matrix with a low viscoelasticity, and you can see how the tissues maintain this spherical symmetry, and there is no significant tissue growth. And on the right side, you can see that in this matrix that has a high viscoelasticity, you can see how the spheroid is breaking symmetry. There is significant tissue growth, a fingers formation, and even some single cells are able to escape the spheroid and migrate. So we define the behavior on the left side as a morphologically stable behavior, and we define the one on the right side as a morphologically unstable behavior. And if we look at the evolution of these experiments that they last 5 days, you can see how in the elastic matrix, they maintain more or less the spherical symmetry. They slightly grow, but if you compare them with the rolling or the behavior in the viscoelastic matrices, you can see how after 2 days, they break the symmetry, and you can even see some finger formations, and they continue to grow even past these 5 days. If we quantify the areas of measurement of the tissue growth and the circularity as a measurement of how spherical they are, you can see how the tissue is continuously growing and how the circularity is decreasing spheroids in viscoelastic matrices. But you can also see that the spheroids in elastic matrices, they maintain more or less constant circularity value. So with these results that we observed that there was an increase in tissue invasion, what we did is first look at potential epithelial to mesenchymal transition due to viscoelasticity, and we observed an increase in vimentin expression in branches in viscoelastic matrices. And we also observed a decrease in cytokeratin 14 expression in viscoelastic matrices. Apart from this, we performed flow cytometry experiments to look at different EMT master regulators, and we observed an overexpression of snail, slug, and ZEB1. Then we performed some in vivo experiments. So these were two different sets of experiments. The first one was with MCF10A cells. So we first developed the spheroids. We encapsulate them in either an elastic or a viscoelastic matrix. We introduced through surgery to mice, and after 7 to 9 days, we removed the gels. And if you look at the results, you can see that cells in the elastic matrix, they do not grow significantly, but you can see how in the viscoelastic matrices, they are able to grow quite a lot. And we could also observe vimentin expression and also slug positive cells, an increase in viscoelastic matrices. The second set of experiments were performed with MDA and MB231 cells. This is a malignant breast cell line. What we did is we subcutaneously inject these cells inside elastic or viscoelastic gels. And you can also see how they are growing much faster if they are inside the viscoelastic matrix. Then we look at mechanotransduction. So we were seeing that there was a huge increase in tissue invasion, so that there was an increase in cell migration. So we thought that there could be differences in the mechanosensitive machinery. So we first look at focal adhesions, and we saw an increase in phosphofocal adhesion kinase in spheroids in viscoelastic matrices, and we could even see some focal adhesions in the leader cells in these branches. Then we look at JAP, which is one of our favorite proteins, and we indeed observed that there was a JAP nuclear translocation in branches in spheroids in viscoelastic matrices too. On the other hand, we could see that JAP in elastic matrices, in spheroids in elastic matrices, they remain in the cytoplasm. So with all these results, we talked with Maha and tried to see if we could develop a physical model where we could predict that viscoelasticity could trigger morphological stability or instability. And Anupam Gupta developed a really nice 3D model where we could predict this behavior. And the model is based on two very simple concepts, which is that first we have like a matrix phase, which is viscoelastic and passive. And then we have a tissue phase, which is viscous, and cells can migrate and proliferate. So the main idea here is that if you have an elastic matrix and a cell is pushing against the matrix is going to push back as the stresses are not going to relax. However, in a viscoelastic matrix, when the cell is pushing as the stresses relax, it's going to be easier to maintain that space. Then we try to validate this model by doing different cellular experiments. I will just show you like a few, which would be, first we look at cell migration. So if cells are unable to migrate, we don't see any kind of symmetry breaking or tissue growth. We perform several experiments. One of those is to use matrices without RGD ligands, so the cells cannot grab the matrix and migrate. We observe that in this case, there is no tissue growth or symmetry breaking. We also wanted to see what were the molecules that could regulate this cell migratory response. So we look at different molecules, mainly focused on the actin, the myosin, or the ion channels. And we first looked at actomyosin contractility by looking at non-muscle myosin 2 or ROC. We also looked at actin polymerization, looking at RAC1 and ERP23 complex, and also we looked at ion channels. Here you can see the results. So we see that RAC1 and ERP23 complex really regulate tissue growth and symmetry breaking. So when we inhibit these molecules, we really see an impairment in tissue growth. And another interesting result that we found is that these drugs do not affect tissue growth in elastic matrices. So it seems a viscoelasticity dependent behavior. If we compare these two molecules and the model, it makes sense that these are responsible for cell migration. So in the model, what the cells can do is push against the matrix to try to generate some space. And these two molecules are known to be responsible for cell protrusion. So then we move to try to understand the influence of cell proliferation. So if there is no cell proliferation and cells cannot divide, there is no tissue growth. So we blocked cell proliferation and tissues were not growing. And another prediction of the model was that if you generate space, it's going to be more likely that cells are going to divide as the stresses are going to be lower. So the idea is that in a viscoelastic matrix, as cells are migrating and pushing, they are going to generate more space. If there is more space, it's going to be more likely that those cells are going to divide. And a similar pattern has been observed in other systems. In this video, I will show you something like this. So these are MDCK cells, MDCK Fuji cells, a monolayer, and these cells are not dividing because they are at a very high density. When they are able to start migrating and they are able to generate space, you will see that they will start to go through the cycle again and start to divide. So we were expecting that in the spheroids in elastic matrix, proliferation was going to be very close to the edges because these are the area where they are able to generate some space, but there was going to be a much broader distribution in viscoelastic matrices. And indeed, this is what we observe. So you can see how in the elastic matrices, we can only see EDU positive cells next to the edges, whereas in the viscoelastic matrices, there is a much broader distribution. And the model predicted a similar behavior. And in the context of the model, this can be explained by an increase in the stresses in elastic matrices. Then we moved and studied the role of stiffness. So this was a very important question for us. And there were very interesting predictions coming from the model. So if you look at the inelastic matrices, independently of the stiffness of these matrices, the spheroids were going to be unable to break symmetry and invade. And in the case of viscoelastic matrices, there were two very interesting predictions. The first one was that spheroids, even in the soft matrix, they were going to be able to break symmetry. But the other prediction was that in viscoelastic matrices, there was going to be an effect of stiffness. And it was going to enhance tissue growth, fingering, and symmetry breaking. So if you look, for example, at the size of in the viscoelastic soft matrices of the tissues, you can see that it increases in the stiff viscoelastic case. So we look at what happened in vitro with our experiments, and you can see how in the elastic cases, they more or less maintain this spherical symmetry and they don't form fingers. And in the viscoelastic case, you can see that in the soft case, the spheroids are able to break symmetry and form these fingers. If then you look at the effect of if you look at the effect of stiffness, you can see how in the viscoelastic cases, stiffness really enhances tissue growth. And also there are more proliferative cells in the stiff viscoelastic matrices. Apart from this experiment, we have done a nanostring experiment, which is a bulk RNA sequencing experiment, looking specifically at cancer pathways to see if stiffness and viscoelasticity were playing this kind of important role. And if you look at the elastic matrices, you can see that the stiffness doesn't seem to activate cancer pathways. However, in the case of the viscoelastic matrices, stiffness really has an effect in spheroids response. So stiffness activates these cancer pathways, but if these spheroids are in a viscoelastic matrix. With all these results, our experimental results and the modeling results, we developed a three-dimensional phase diagram to try to predict when a tissue is going to be morphologically stable and when it's going to break symmetry and form these fingers. And this is dependent on three-dimensional parameters. So the first one depends on cell motility and material properties. So if the cells are more motile, it's going to be more likely that they are going to be morphologically unstable. And also if the matrix is more fluid or it's stiffer, it is also going to be more likely that they are going to break symmetry. The second one is related to the relationship between the tissue viscosity and the matrix viscosity. So if this ratio increases, it is also going to be more likely that the spheroid is going to be morphologically unstable. And the third parameter, it's related to cell proliferation and how proliferative your cells are, or the spheroids are. Next, so I have shown you all these results related to the breast spheroids and how we can control or we can predict when it's going to be morphologically stable or unstable. And we wanted to check if in another system it could happen the same thing. So we decided to look at intestinal organoids and see if viscoelasticity could trigger symmetry breaking, tissue budding, and tissue patterning. So indeed, we observed that in viscoelastic matrices, the organoids are able to break symmetry and grow. However, in the elastic matrix, we also observed that they maintain this spherical symmetry, even if they are able to grow with time. If you look at the quantitative results, you can see that there is a similar behavior to what we have observed with the breast spheroids. We also observed that viscoelasticity enhances the number of organoids with stem cells and also the number of cell colonies that are able to grow with time. And we also look at the development if the organoids are able to differentiate in these matrices that we are using. And so we have seen that there are Paneth cells, and we more recently have seen that there are endocrine cells, goblet cells, and enterocytes in our organoids. And we also look at the effect of the stiffness. So here we weren't sure what we were going to find, as these organoids are usually cultured in matrigel and it's a very soft matrix. But indeed, we find that when we increase the stiffness of our matrices, the organoids are bigger and they have more proliferative cells too. So it seems to go in the same way, or it goes the same way as our breast spheroids. So as a conclusion from these projects, I hope that I have convinced you that the extracellular matrix viscoelasticity regulates tissue organization. I have shown you examples of symmetry breaking, tissue branching or budding, tissue growth and patterning, both in breast epithelial cells and intestinal organoids. I think that these results explain partially previous results, that when a synthetic material that is purely elastic, if you culture an spheroid inside this material, they are not going to break symmetry and invade. However, if you use a natural matrix, you would see that at the same stiffness, they are able to break symmetry and invade. And apart from this, I want to highlight that there are already meaningful examples of the role of ECM viscoelasticity in health and disease. There is a very nice work from Celeste Nelson's lab where they show that ECM fluidity increases during lung development in chick embryo. And also there are some examples that have shown that in cancer, tissue viscoelastic properties increase in brain tumor compared to brain parenchyma, and also in breast and liver malignant cancer if you compare them to benign lesions. So I think that also that these new results open new avenues to study the role of matrix viscoelasticity in different processes. I would like to end by thanking first the Mooney Lab. So most of this work has been developed in Boston, in Dave's lab. I really want to thank Dave for all his help and support, and all the people involved in this project. I also want to thank Maha and Anupam for the amazing model that they have developed and all the hours that we spent trying to figure it out, and also the Zoe Lab for their help with the organoids. I just started a few months ago my lab here at the Creek in a joint appointment between the Creek and King's College. And Olivia has started in the lab, and Eduardo and Brenda will start soon. And we are currently hiring postdocs, a senior laboratory research scientist, and a PhD student. So if anyone is interested, please contact me. And thank you very much for your attention.
Thank you so much, Alberto, and congratulations for your new lab. It's so exciting when that happens, right?
Yeah.
We are looking forward to what you're going to do in your lab and the future of it. I'll take a few of the questions. So the first question is from Kinjal. She's asking, in the physical model, is the cell migration rate assumed to be mechanical stress dependent?
Well, the cell migration, we are changing it with the stiffness. So it changes with the stiffness, but it's not stress dependent. What is stress dependent is the cell proliferation. So if the stress is higher, it's less likely that the cells are going to divide.
All right, great. From Kasanan says, thank you for your talk. Did you observe any change in matrix viscoelasticity after the branching? And then a second follow-up once you answer that.
So we haven't been able to check the viscoelasticity. You mean in the bulk gel? So in the bulk gel, we haven't seen any big changes not significant changes, but it's true that in our materials, we don't know when the cells are pushing and generating some space, if they are changing with time the properties of the matrix. That's a very interesting thing to look at.
Yeah, I think that'll be great to figure out. And then the follow-up question is whether the viscoelasticity is allowing the spheroids to branch, or is it triggering the spheroids to branch?
Well, I'm not sure what it is. I would say that they are allowing the spheroids to branch. So I think that the cells are trying to push, and in the case of the viscoelastic matrices, they are allowing to generate the space to invade. Whereas in the elastic matrices, when they are trying to push, they cannot generate that space, and then they form these spherical symmetry tissue.
Thank you. Next question is sort of along the same line from David, where he's asking regarding sensing of the matrix viscoelasticity, what is known regarding the role of traction versus pushing forces?
Well, there are some papers published by Obi's lab, the Chowdhury lab, Senoy's lab, where they have used the molecular clutch model to try to predict how cells behave in viscoelastic matrices. But I think that even if you are trying to use the molecular clutch model on a purely elastic material, it's unclear what happens when the cells are pushing. So we are not completely sure how you could combine the pushing versus the stiffness sensing mechanism. So I am not sure how that works.
Okay, fantastic. Next question from Raphael Petrosian, who we can always count on for great questions every week, says that single cells themselves can be viscoelastic. So what fraction of the tissue level viscoelasticity do you think arises from individual cell viscoelasticity? And what fraction from the cell-cell interactions and the extracellular matrix?
Well, so we think that for our systems, what we were looking at, and we try to make some measurements, even if they are not included here, of the bulk viscoelasticity of the tissue to see if the relationship between the matrix and the tissue. And I think that that's interesting when the tissue, for example, is pushing against the matrix. If you are looking more at a single cell behavior and how they are able to migrate or navigate through the matrix, I think that it makes more sense to look at the single cell viscoelastic properties.
Thank you. Next question is from Amelia. She says, great talk, Alberto. Could you speculate if basement membrane are more elastic or less viscoelastic compared to the stromal ECM, and if this might contribute to epithelial tissue homeostasis?
Well, that's something that I want to look at. So we have started to do some measurement, but to be honest, it's completely unclear. So what I would expect is that the stroma will be more viscoelastic, so that the cells will be able to navigate and invade in those cases. But there are many things to do also. So one thing that we want to see is, for example, the collagen that is formed around the tumors, that it seems to limit how the cells are able to invade if that's less viscoelastic, and that's preventing the invasiveness. And if you are able to cross that space, then it's going to be much likely that tumors are going to be able to grow. But we don't really know nearly anything about that.
Awesome. So it sounds like you have a lot of work to do. So we're approaching just the end of the hour. Are you okay to stay on to ask a couple more questions or answer a couple more?
Yeah, of course.
Okay, great. So from Natasha Golovchenko, you've already touched on this a bit, but she wants to know about cell matrix interactions by which the migrating cells are influencing the viscoelasticity of the surrounding matrix.
Can you repeat it? I didn't understand it well.
Can you talk more about the cell matrix interactions by which the migrating cells are influencing the viscoelasticity of the surrounding matrix?
Okay, so, well, there are a couple of papers that they have checked, they have looked at matrix deposition when the cells are migrating, and they have seen that the matrix that cells deposit is more fluid-like than the surrounding matrix. So one interesting hypothesis that the cells are like depositing some kind of viscoelastic matrix that is favoring their migratory path. And as how that happens, I think that we don't really know exactly how it happens. So we could use the same cell matrix interaction molecules that we have used to understand how cells sense the stiffness to apply them to viscoelasticity. And so far, it seems that similar molecules regulate both responses.
That sort of also answers Natasha Buer's question. So I'll take another question from Jeelan. She said, I'm curious, how does the viscoelastic time scale affects the results?
Yeah, so we don't really know. So it's some on 2D there is some work done that they see that there is like an optimal time-dependent property that allows cell spreading, for example. But in 3D, we don't really know how it will affect. So far, from our results, having a faster stress relaxation, it would favor invasiveness in all cases. But I don't know if there is an extreme case that if it's too fluid, that the cells are not going to grab the matrix and invade. So that would make sense to me, but we haven't found it.
All right, thanks. Our next question is from Jim Butler. So he's asking if the only functional difference between elastic and viscoelastic is a difference in time scale, like the difference in the relaxation time, then would the two movies look the same if you replayed them at the sort of the same ratio as the two time scales to each other?
Well, in the model that we have, we allow also for plastic deformation, so it would not be exactly the same. So it would be something different between elasticity, purely elastic, and a viscoelastic case. And if the deformation is too, it goes above a threshold, it will become a plastic deformation, a permanent deformation that would favor invasiveness. So I wouldn't say that that's the only difference between both movies. So there is another one which is quite important, which is that there can be plastic deformations.
So if you left the elastic long enough, it wouldn't turn into the viscoelastic case.
So it would also, the cells should be enough to generate a deformation high enough to generate that plastic deformation. If not, you wouldn't see something similar. And the ability to generate a plastic deformation, it will depend also on the molecular weight of the alginate. So it's going to be harder to reach that deformation for the cells to invade. So I don't think it would be the same case. I understand what he's trying to say, but if you don't add the ability, if the cells don't have the ability to generate that plastic deformation, you won't see the symmetry breaking and invasiveness.
Thank you. The next question is more sort of technical. From Sangyun, how did the model predict proliferation at the tissue periphery, and what was the condition in the model to sort of allow the proliferation?
So the proliferation, it was like it depends on a probability equation, and the probability increased when the stresses were lower. So if the distance between cells increased, the stresses were going to decrease, and that was going to lead to a higher proliferative rate. So the cells will be able to generate more space when they are closer to the edges. That's why we observe like more proliferation on that area.
All right, we're skipping over a bunch of people saying fantastic job, but we'll send you that later so you can see all the compliments. A question from Camilla Thurudi. Do you think the presence of vasculature in the organoid would change the behavior that you observed from the viscoelasticity point of view?
Well, I don't think that would change. I think that with the results that we have, that it favors branching to try to combine endothelial cells with the organoids. It could even promote the growth of the organoids even more compared to the elastic cases. So we would have one system where the endothelial cells cannot form these branches and the organoids cannot grow, with another one where the endothelial cells will form these sprouts and maybe connect with the organoids and even grow more. So I don't think that, but I haven't tried, so I don't know exactly what would happen.
Great. So the next question I'm taking is from Sebag. Do you think that the viscoelastic matrix breaks simply cell confinement or interaction with matrix viscoelasticity is involved. Sorry, sort of
Can you
muffled it.
repeat it?
Yeah,
Sorry,
I muffled it. It's my fault. Do you think that the viscoelastic matrix break simply self-confinement, or interaction with the matrix viscoelasticity is involved?
Well, I think that, well, that's a good point. So in our case and our matrix, the cells have to generate the space to invade. So there, you need to have that. So there is a confinement in that sense. So when they are able to displace the matrix, they are able to invade. If it was like if there were like huge pore size, I wouldn't expect that the cells will behave the same way, and the differences will be closer to. I would expect that it will be closer to what it has been shown on 2D. So yes, I think that the confinement plays a role in our results. Yes.
All right, awesome. I think we're getting close to the end here. From Giorgio Skita, says very neat talk. And then one of the key parameters of your 3D phase diagram is the ratio between the tissue fluidity and the matrix viscoelasticity. So because that ratio is important, have you tested experimentally the impact of changing the tissue fluidity?
We are doing that now. So we are trying to change the viscosity of the cells and see what will happen in our system. What I would expect is that if the rate increases, it should promote more branching, but we don't have the results yet, so we haven't done that.
I'll take maybe one last question from Pradeep. He says, what happens to the model if it is run for a longer time? Is there a feedback considered between the matrix and the cell? Do you expect similar behaviors in 2D?
So related to the first part, so there shouldn't be any changes on, so the spheroids in the elastic matrix, they will maintain like the same shape. And in the case of the viscoelastic, they will keep growing. Yes, so that would happen for sure. On 2D, I would expect that it would be something completely different, but because from what I responded before, that in 3D there is this confinement that the cells have to displace the matrix. So I would expect like that cells will be able to grow and migrate in both elastic and viscoelastic matrices as it has been seen before.
All right, we actually, we've got a question that I hadn't noticed from YouTube asking if the pore size of the hydrogel matrices is, I guess, important.
Well, the pore size is it's the same. So in previous work with this alginate, they didn't find any differences between the different molecular weights. As if it is important, yes, it's relatively important, because if it was big enough, then the cells wouldn't need to displace it. But also say this, I think that also it's important to notice that many papers in the literature talking about cell migration in 3D, they thought that if the pore size is smaller than like the cross section of the nucleus, the cells are not going to be able to migrate. And I think that not only this work, by previous work, they show that if the matrix is viscoelastic enough, the cells are going to be able to displace the matrix and migrate, even if the initial pore size was smaller than the cross section of the nucleus.
I keep saying last question, but there's one more.
Well, no problem.
I wanted to ask if sorry, it's from Sudeepa. So she wanted to ask if the proliferation within the cluster increases extremely, would there be a critical point at which the cluster would be able to break symmetry, and even on an elastic matrix?
So we haven't performed like for the breast spheroids longer than a week. So I don't know after a week if it could happen. I wouldn't expect that. So even the experiments in Vivo do not look like that. We have increased even more the stiffness to see if it could lead to symmetry breaking, but we haven't. I wouldn't expect, so I already start with a quite big spheroid. So, but I don't know if there could be other kind of signals after some like after a week or 2 weeks, that could also play a different role apart of mechanics. But I wouldn't expect that.
I think that's the end of it. Thank you so much, Alberto. That's just wonderful.
Okay, thank you for the opportunity. I'm
Yeah.
glad that I was able to present.
Yes, and especially both Adam and Alberto, you guys did this on really short notice and let us sort of kick off the spring or fall, you know, with a really great seminar. So thanks to both of you. I think we're actually maybe we can return to a few of the questions that we didn't get to for Adam. So a bunch of people have left who left questions, so we won't go through those. But I will return to Giorgio's question. Adam, are you still here? Oh, there you are. Hi. Great. So Giorgio had asked, actually, you know what? That's not a full sentence. I'm not sure. Giorgio, if you want to jump in and ask your question, I'm not sure what you were asking there. So the next one from David Lagares says, developmental programs are reactivated during fibrosis and tumor metastasis. Do you think that the durotactic mechanism you identified in Xenopus might play a role in disease?
Yeah, definitely.
Yes.
I'll expand. I mean, there's at the moment, there's, like I said in the talk, that there isn't much known about Durotaxis in vivo, but everyone thinks that fibrosis is the most likely place that you're going to see it, because what you want to get an efficient Durotaxis is a really steep gradient, because steep gradients are better than shallow gradients. And the way you get steep gradients is by tissues stiffening much more than their physiological sense. And that's what happens, you know, in cancers and in fibrosis. There was a study back in 2005 where they measured the stiffness of some fibrotic hearts, and they showed that the gradient gets really, really steep. So it's the most likely place you're going to see it is somewhere where there's a steep gradient, and fibrosis is probably the best bet for that.
Great. There's another question from Sebag. The embryo has several layers, so how do you discriminate between cell stiffness and matrix stiffness?
Yeah. So I mean, I didn't mention this talk, but in order to access the placode isn't the most superficial tissue in the embryo. So you actually have to take off the superficial epithelium. We did a bunch of controls for that to check that wasn't affected. Then once you take off the superficial epithelium, if you then fix the embryo and you stain it for placode and ECM, there's no ECM at that most superficial layer. So we know that when the cantilever is coming down and hitting the placode, it's not really hitting ECM. And we don't think it's hitting the ECM on the other side of the placode because a previous paper from the lab has looked at that, and in comparison to the tissue, it seems relatively soft. Additionally, the fibronectin in the layers are extremely thin in comparison to the layers of the tissue. They're like 1 micron. And we don't do indentation so deep that we might be picking up mechanical properties of stuff that is too deep. So we think it's the tissue.
All right, of the YouTube questions, or not YouTube, of the Zoom questions, I think everyone who's still here, we've gotten all of theirs, and the ones that we didn't get to are people who left. So we'll copy that for you and we'll send both of you the list of questions so you have that. But then one more from YouTube from Nath House asked, when you create the ectopic neural crest cell migration, is their location at all affected? I'm not sure which part that was in reference to, though.
So when we make the neural crest go in the opposite direction, then yeah, they're in an ectopic location. I'm not sure. I mean, it depends where you push. So if you push it in the direction where they should go, then they are where they should go, and if you put it in the other direction, they are where you don't expect them to go.
Adam, I think you did get to this to some extent, but I'm curious, just if you think about competition between the cues? Do you have a sense of, because I know you said you need to do both reversing the gradient and doing the pushing to get the cells to move in the wrong direction. But I'm just thinking about, do you have a sense of like what's stronger, chemotaxis versus durotaxis, or does that not really apply in this situation?
I don't know how you can answer it. That's the problem. How do you compare?
Well, we can do it in vitro.
Can you? I mean, how do you compare a gradient of 1 millimolar chemical to 1 kilopascal stiffness?
Okay. I don't know how you compare apples to apples, but you could put different ones of sort of different strengths in competition with each other.
Yeah. So I would fully expect that if there's a chemical here and the stiffness is over here, and you increase the chemical, then it's going to be, you're going to bias it to go to the chemical, and if you increase the other one, it's going to be biased towards the other.
So there's probably a phase space.
Yes. And probably it's dependent on the stiffness and of that phase.
Yeah. I guess there's probably,
and the chemicals, right?
Not gonna, which ones? It's not gonna be an easy answer. Just thinking about what we should do next. All right. Ankita, I think there's one more if you want to ask that.
So it's from Haoyang. Sorry if I'm pronouncing it wrong. Could cadherin interact with each other through fibronectin? It seems in adult tissues, basement membrane only allow integrin binding, but not cadherin.
So we think the protrusion is going through the holes in the fibronectin to hit the placodes. So we already know from published papers that the placode and neural crest are interacting into a cell-cell interaction, which is the NCAD. And then separate to that, the neural crest is also interacting with the fibronectin, which is through integrins. So we're certainly not proposing that the cadherin is a means of interacting with the ECM, which would be super cool if it was true, but I don't think there's any evidence for it.
I think that's it, right? We covered all of them.
Yeah, I think so. All right.
Yippee!
Fantastic. Thank you both so much. And everybody, we will